Hello, everyone. My name is Adrian Scheibler. I'm here from Telluride. And I spent my summer at the Crawford Lab, which is part of the NorthShore University HealthSystems Research Institute in Evanston, Illinois. So our goal at the Crawford Lab, we study the prostate organ and its numerous diseases. Specifically, the project that I worked with was with finding the relationship between obesity and prostate cancer. So what we know is that if you are obese, you have a higher likelihood of dying from disease, and obesity is a significant risk factor in the progression of prostate cancer. However, it's currently pretty controversial of whether obesity influences the onset of prostate cancer. So the project aim was twofold. For one, we wanted to find out the location and the expression of a certain transcription factor called GATA3. And we also wanted to test whether GATA3 location was altered when we applied a lipid challenge of oleic acid, which is meant to mimic obesity inside the cells. So GATA3 is this actually like really cool protein that has a lot of different functions, and it's mostly well known in the kidney and especially in breast cancer. In breast cancer, it's a major regulator of differentiation, and loss of GATA3 is actually a really poor prognostic indicator. So the loss of that protein in breast cancer especially is a very bad sign. And in prostate cancer, the molecule is actually pretty unknown, and there's not a lot of research on it yet. However, there are some indications that's pretty influential. For one, the loss of GATA3 has been correlated with an increase of certain tumor-promoting pathways and loss of certain tumor suppressors. And second of all, the loss of GATA3 has been shown to really lower has been correlated with androgen insensitivity and castration resistance, which really makes one major form of treatment against prostate cancer very inefficient, and which really makes the cancer much more aggressive. And finally, the GATA3 is pretty important in the mitotic spindle process. So it kind of organizes the whole microtubules that physically separate the chromosomes. And upon loss of GATA3, this whole process is really randomized. So we don't know how the cell division is going to work out, and that can lead to some major problems. So this is a really cool picture that I took with immunofluorescence. So that was the first kind of major thing that we did. And in it, we gave 2 rounds of antibodies to the cell, which target the different proteins inside the cell and make them light up under the UV light. So the green inside the nucleus mostly is the GATA3 protein, and then the red is GM130, which shows up where the Golgi apparatus is, which you can kind of think of as the post office of the cell. And as you can see here, it's a lot of expression inside of the nucleus, not as much in the cytoplasm. The second major test that we did is Western blot, which Joey explained in her presentation, but it pretty much attempts to quantify the number of proteins in a given cell sample. However, over the 3 weeks that we tried this method, all of the tests failed for whatever reason. And you can actually see this down here where there's a missing column, which is bad. And here where it's just too wide, and the gel that we used for gel electrophoresis was also just bad. So we had a string of bad luck, but that's what happens in research. So some other really cool things that I found out in the lab was that we use milk powder, which, going into it, you just don't really expect. We looked at some prostate cancer tissues, and they let me use this really expensive microscope, which I was pretty scared to handle, honestly, because it's expensive, and yeah, it's easy to break stuff. So we actually had a really cool surprise finding that generally got us really excited, because the way, cancer is really uncontrolled cell proliferation, so the uncontrolled mitosis of cells. So when we looked at these immunofluorescence samples, we found that there were a ton of mitoses on the slides. And we found that the GATA3 seemed to colloquialize, so show up in the same places as what's known as Aurora B, which is a kinase that orchestrates the cell division and helps with the microtubules. So what made us even more excited, however, was that there seemed to be a difference between the treatment group of oleic acid and the control group. And I mean, this is really, I mean, this kind of groundbreaking was what told to me, because we really had never seen this before, and we never really knew that there was an association between those two proteins in this kinase. And this is probably my favorite picture that I took of my entire time there. And I mean, I have a ton of pictures. I have a couple of gigabytes of them. So like, this is my favorite of all time, because what happens in this one is the spindle fibers are coming right out of the page. And so you have this really cool shape that is formed. The blue is actually the chromosomes, and then the red is that aurora B that we were talking about earlier. And you can actually kind of see the colocalization a little bit in, for example, in this area and this area, where it kind of forms a yellow as the green and the red are aligned on top of each other. So Chicago, I was in Evanston, Illinois, which is just north of Chicago, and it was a really cool time. So I stayed with 3 different host families. The first and the third were related, the Steinbachs, and later on with their son, his family, and also the Adamolis. And they took me to really cool places. They introduced me to deep dish pizza, which, I mean, I'm going to admit is a pretty acquired taste. And I went to a lot of concerts, like I think this is Red Hot Chili Peppers. I went to the art museum, which kind of became like a second home. I went to Wisconsin a bunch. Yeah, that was pretty cool. I went up to the Sears Towers, and it's really amazing just to see this pretty much a sea of concrete in front of you, the bean, and so going around Chicago, exploring different sites there. Also, Dunkin' Donuts is really good. I'd never really been to Dunkin's before, but yeah, America runs on Dunkin's, and so did I. Yeah, and lastly, thank yous to everyone, and yeah, thank you.